Sure, yeah. Quick break, I'll pull up the presentation.
Yeah. No, actually, I'm just graduated right now, so I'm just working full time.
Oh, okay. Okay.
Yeah.
That's what I was wondering.
Yeah.
Okay. So not eligible.
Yep. So not grading.
All right. Sounds good. Okay. So we are going to do our last presentation here. Madison Heater from Gannon University. Not a student, so you don't need to judge this presentation. But looking at factors influencing COVID-19 spread on a university campus setting. And with that, I will let you take it away. And then you don't use the
Yes,
card.
thank you. To make sure. Oh no, I just messed it up. See?
Oh, I think you're good.
There we go. Got it. I'm not used to fancy technology when I get to present. Don't blame me if it's bad because I practiced on my dog and she's not a very good. She can't give me much feedback. So, hello everyone and good morning. I'm very happy to be with you all today in your lovely shining faces and happy Friday. As you guys all know now, my name is Madison Heater. I'm the COVID-19 lab technician and research assistant in the lab at Gannon University. I'm honored to be with you all today representing Dr. Stephen Morrow and our lab crew. All right, so the elephant in the room, COVID. Coronaviruses are a family of viruses that infect humans and animals. And this family of viruses includes SARS-CoV-2, which causes the disease COVID-19. Coronaviruses can cause colds and in some cases, severe respiratory infections and diseases. All coronaviruses have a single viral genome of RNA surrounded by an envelope. You can see here, so they're in here. And corona means crowns. So there's a crown of protein spikes sticking out of the envelope of the virus, which you can see right there. And these allow the virus to attach to specific proteins on the surface of cells within the body to fuse with the cell membranes and release the virus's RNA. The new form viruses are eventually formed and released outside of the cell and can infect other cells or be released outside of the body via droplets or mucus. Once in humans, the virus mutates not to create an entirely new virus, but enough to create variations of strains of an already existing virus. There's no doubt that COVID-19 has changed the way we live our everyday lives. Basic practices in the workplace and within higher education across the United States have been altered in an attempt to mitigate the effects of COVID-19. So just a little bit of timeline for you. Looking back to 2020, which seems so far, but yet so close. When this pandemic first started in the United States, I want to highlight a little bit about what we saw during that time. January 9th of 2020, WHO announced the coronavirus-related pneumonia seen in Wuhan, China. Come January 21st, 2020, the CDC confirmed the first US case. Not too long after, February 5th of 2020, on Gannon University's campus, we were closely monitoring the outbreak and what kind of effects that may have to our campus. March 17th of 2020, Gannon University was forced to close the Erie and Ruskin campuses. Come August 6th of 2020, it was move-in day for the class of 2024. In order to come back in the fall of 2020, we had to have a few different protocols in place in order to do so. So things looked a little bit different on campus for the fall and spring semesters of 2020 and 2021 as a result in the effort to mitigate the effects in the spread of COVID-19 on campus. Face masks were required everywhere on campus. Signage was posted to reduce traffic, and you were only allowed to enter and exit in certain doors of buildings. There were limited class sizes, and classrooms were readjusted to encourage social distancing. There was mandatory sanitizing before every single class. Remote delivery options were available as they were needed. There were temperature checkpoints in many entrances to the buildings. There was a Live Safe app, which is pictured here, that was used in order to monitor symptoms of students and staff and was required every day before attending class. And of course, our COVID-19 testing was implemented, where students were pulled at random for testing, and our student athlete population was tested in accordance to NCAA rules based upon the risk status of their sports. These were ranked according and assigned by NCAA and were ranked from extremely low to high risk. So for example, usually swimming was considered extremely low risk for COVID-19 versus wrestling was extremely high risk. So, come fall 2021, we have seen a couple protocol changes. So first, throughout the sector of the summer on Gannon's campus indoors, we weren't required to wear a face mask. That changed pretty fast on August 10th when we had to wear a face mask again indoors. The Live Safe app is still used every morning and required in order to monitor the mental and physical health of students and staff on campus. Gannon is not requiring that vaccinations have to be taken before coming back to school, but they are strongly encouraged. We started the school year at about a 50% vaccination rate for students, and we've seen an increase in those rates both on the Erie and Ruskin campuses. Right now on the Erie campus, we sit at about 73% of the total student population being vaccinated, and at the Ruskin campus, about 85% of the student population is vaccinated. We also are having vaccine clinics at the university on campus in order to allow access for students and staff to get their first or second dose and booster shots. And of course, we still have our COVID-19 testing on campus. This year looks a little bit different as to who we are focusing our testing efforts on. The last 2 semesters, our bulk of our testing focused on the student athlete population, and now we are doing surveillance on staff and students who remain unvaccinated. They're tested bi-weekly, and once vaccine rates reach about 80%, Gannon's Erie campus will reevaluate our surveillance program and the testing on unvaccinated individuals. The online dashboard and vaccination reporting systems are still in use these last 2 semesters to highlight the campus-wide data, such as the number of students and staff in isolation due to infection. However, we did lift the temperature screenings for students and staff, lifted gathering limitations, and classrooms and laboratories are now allowed at full capacity. So you may be asking yourself, okay, you told me a little bit about what your campus does, but why does this matter to me? Well, ladies and gentlemen, that's an excellent question. We are only one of the universities in the area specially developed an in-house system where we test our students and staff and have results back within 8 to 12 hours after the specimen has been collected. Specimen is collected via nasopharyngeal test and stored in viral transport medium and refrigerated. These samples are then vortexed for 10 seconds, transferred into Eppendorf tubes, and boiled for 5 minutes at 95 degrees Celsius. We use 15 microliters of master mix and 5 microliters of resuspended sample and add it to wells for PCR. An applied biosystem 7500 platform with thermocycler is used to amplify the genetic material. So you can see that here. It's our machine. We have 2 of them, luckily, because otherwise I would get nothing done. qPCR looks at the quantification of amplification through each PCR cycle, and the reaction progresses via fluorescence signal. We are utilizing our qPCR to amplify genetic material from the samples from the students and staff. If the viral genetic material is present, the signal is produced using a fluorescent probe, and we're able to tell based upon the control standards if the viral load is present and if we can therefore call a patient positive. So here you can see our standards, and this yellow curve right here would be someone who's considered positive for COVID-19. When looking at cases of COVID-19 in Pennsylvania and in Erie County, you can see an overlap between the percentage of positive cases in the county in comparison to the overall positive cases in the state, as shown here. But then, when you look at Gannon University compared to Erie County and Pennsylvania as a whole, we're not seeing the same trends. Our data suggests that trends of positivity rates on campus are not largely due to difference in daily testing volumes, but rather they arise from unique features of campus community, which cannot be easily projected by statewide or even community-wide trends. So as you can see here, this is the county positive, this is the state positive percentage, Gannon is down here, and we are seeing significantly different trends between the two. Although Gannon adopted national and state guidance and enforced other measures, including rapid surveillance programs and testing, in an attempt to mitigate the spread of COVID-19 on campus, we are able to limit but not eliminate the occurrence of outbreaks on campus. Although the adopted measures are essentially necessary for in-person instruction, they're not sufficient in containing the spread of COVID-19 completely on the university's campus. A paper that we just published in the International Journal of Environmental Research and Public Health, where we discuss our results that demonstrate that student athletes are a major drive of COVID-19 infection within the university campus environment, highlights this here. In summary, we found that enhanced surveillance focused on the entire student athlete population, and it's recommended that routine testing of all student populations is conducted if the resources are available in order to properly monitor the spread of COVID-19 on campus. Ultimately, it is our goal to maintain a testing positivity rate below 2% campus-wide to maintain in-person instructions. Luckily, as you can see here, our testing positivity remains below, maybe if I can get there, below 2%. In comparison to Erie County, which is a testing positivity rate of 23.9%, and statewide, which is 8.4%. Now, looking forward to what we're going to be doing in the future, I want to talk a little bit about the research that we've done and we're looking to do. Dr. Stephen Morrow and Austin Hertel, a senior student at Gannon University, are my 2 main colleagues that work alongside me and work at the research front of our lab. Unfortunately, my colleague Austin Hertel was supposed to be with me here today, but he was unable to make it due to his student-athlete status. So I'll be speaking on his behalf, but I do encourage you to direct any questions to Dr. Stephen Morrow or Austin Hertel about our research front. One portion of our research is focusing on metagenomics. We are studying the relationship between gut dysbiosis or distribution of microbiota homeostasis due to an imbalance of mycoflora and COVID-19. We're utilizing next-generation sequencing, or NGS, to establish a relationship between gut microflora, and infection with COVID-19. So far, it has yielded very promising results. When we compare it to what would be considered a normal gut microbiota, we see a certain distribution of many types of microbiomes found within the gut. The data suggests that COVID-19 has a major disruptive effect on the host microbiota during infection that persists long after the infection is clear. So here you can see when a patient tests positive versus when they're negative, how the microbiome is changing within your gut. Another portion of our research will be focusing not only on influenza A and B screening, but also COVID-19 variant test screening and using real-time PCR to review mutations present in several areas of concern and variants of interest. Our goal in doing so is to monitor the spread and emergence of COVID-19 variants on our campus and throughout the Erie community. And we will be looking at a series of different mutations which are highlighted, not there, sorry about that. Potentially, this research will allow us to create a timeline for when we saw certain types of variants come into the Erie community and on our campus. We'll be using NGS again to reveal insights into temporal patterns emerging of COVID-19 in a university campus setting. So now I want to introduce you to the lab team before I hand it over to questions. This is Mara. She is one of our graduate students. Ross and Gwen are new to our lab. They're 2 undergraduate students. Austin Hertel is pictured here. He is a senior this year. That's me, I'm here. And then a huge shout out to Dr. Stephen Morrow, who's the vice president on campus. He's been such a huge help and has basically brought this program to Gannon to ensure we can keep Gannon and our community safe. So trust me when I say me and my team have had many late nights that have turned into early mornings to work hard to deliver necessary results to the community and research to move forward with COVID-19. Everything we do would be impossible without the hard work that we all put forth each and every day. COVID may have changed all of our lives, and our lab team will be sure to dedicate our efforts to contribute to the science community as much as possible in the months to come, while keeping our community safe with surveillance testing. Thank you.
Questions?
I always have one.
Oh, that's good.
I must be stupid. Did you do any, send any of your samples to external labs to do quality control checks?
Yeah, so that's a great question. Actually, I did bring, if anyone's interested in looking at the paper we published, it is in here. So Gannon University is not considered CLIA certified, so every single time we get a positive test, we do have to send them to, for example, like Hammett or St. Vincent so they can get further testing done and it can be confirmed by a lab there. So yes, we always check and make sure that we're doing great with the work we're doing, so.
So it's pretty consistent?
Oh yes, very consistent,
That's
yep.
good to know.
Yeah, very good.
That data just looks so good that you've got such a low rate.
Yeah, it's awesome to know that our mitigation efforts are working on campus, and we are constantly reevaluating to make sure they are working. A lot of what we're finding is when we are seeing positive spikes in the population, when we're contact tracing, we're finding that it's due to gatherings that don't align with the mitigation effects we have on campus, to say the least. So
People breaking the rules, huh?
yeah, I know. Isn't that weird? I would never expect that. Sarah.
So I saw in your graph where you're comparing it to Erie County, Gannon to Erie County, and then Erie, Gannon to the state.
Mhm.
There's like a little part where Gannon starts to go up, and then it like plummets down and levels off. What time frame is that? 'Cause that just looked like a really interesting.
Very interesting question. That was right before we left for winter break. So luckily, we did not have to worry too much about those kind of issues happening on campus because everyone was pretty much going home, and if they weren't going home, they were able to stay on campus and isolate for the correct amount of time. We got really lucky because otherwise we probably would have had to shut down the university for 2 weeks, and we really didn't want to do that, so yeah.
Do you ever send out the negative test just as, so, and the same thing, you just, they come back negative?
Yep, correct. And sometimes too, so with our standards right here, I only have 3 of the standards pictured, but we do have a 10 to the 4 standard. Usually, if we have a sample that falls between 10 to the 3 and 10 to the 4, we'll call them inconclusive, because we found that either A, that person could be in early stages of infection, or B, they're at the latter part of their infection. So to kind of cover our butts, we call it inconclusive, but also send them to a CLIA certified lab to make sure that they aren't going back into the community and spreading COVID.
The metagenomic surveillance analysis is fascinating, but I'm a little dismayed that it was just one person involved.
So I shouldn't say it's one person, it's the whole lab involved, but mainly we're kind of in the works of it still. That was like our initial findings with it, and we kind of didn't know what to expect, and when we got our results back, you could say there was a little bit of party in the lab because we didn't expect to see such a drastic change in positive versus negative microflora. So we're still kind of in the works of it, and now the whole lab is kind of hands-on. Before it was just Steve and Austin over the summer, but now that we have more people in the lab, between surveillance and that, we're all working kind of together to get that going and hopefully publishing a paper.
I'd be curious, what would be the experimental design for number of subjects and
Sure.
labs?
So the hard part about that is you usually need feces from a positive COVID-19 patient. So I think that's going to be interesting, kind of finding where we can get that. We've had a few individuals from our lab or individuals from our lab who have family members and or friends who are willing to give us feces for the study. But yeah, right now we're working with what we have. Hopefully soon we can kind of get it rolling and see if we can get more involved in it. But that's
Kind of cool to collaborate with a hospital.
it would be awesome to do that actually. That's an excellent point. We might have to look into that.
That's cool. I'll be dying to see the results. I hope. Yeah.
Not literally.
Yeah, not literally.
I'm going to be part of it. I mean,
Any other questions? All right, well, that's the end of the presentation